DNA extraction is, as its name suggests, a technique used to obtain DNA from organic samples, in this case, blood. There are different methods to accomplish this, but regardless of the method chosen, the process consists on three basic steps. Cell lysis. By a lysis solution, which is rich in salt, we break macromolecules such as proteins and allow DNA denaturalization. This process shall be repeated several times to ensure the degradation or disappearance of any other macromolecule. To eliminate the cell membrane, we can use detergents such as SDS 10%. The next step consists on protein fraction degradation release to DNA. We can use sodium acetate to accomplish this. Finally, the purification. Since the DNA does not dissolve in alcohol, we can use cold ethanol and recover the genetic material by centrifugation. The next step consists on recovering the pellet, which contains DNA and it can be suspended in water for conservation. There are several differences between RNA and DNA extraction. The best way to extract RNA is using trace-sol. A reagent is ready for use in isolation of RNA from cells and tissues, and it is not as useful for extracting DNA. While DNA can be stored for a while, the RNA should be extracted the same day that the sample was obtained. And while in DNA extraction we can use RNases to degrade the RNA in the sample, in RNA extraction, obviously, we cannot use this. Column chromatography has proven to be a very useful tool because it has several mechanisms of separation. The modification of the different existing stationary phases as well as the development new is the main basis of obtaining the necessary conditions for the rapid and efficient separation of biomolecules. These modifications or different ways of using column chromatography made of this a very adaptable and flexible process. Some of these are the permeation chromatography, ion exchange chromatography, and adsorption chromatography. To verify the presence of DNA, we can use several methods like spectrophotometry, quanti-blot, which is the quantification method by specific proof hydration of Perkin-Elmer, optic density, and using the Lambert bureau.
Samples did not have a great quality. The ratio from the absorbances gave values below the ones of a pure sample. For DNA, the value from the sample was 1.51 and the expected was 1.7. For RNA, the value obtained was 1.42 and the expected from a good sample is 2. From the DNA, there were 1.510 micrograms. From the RNA, the sample contained 4.925 micrograms. When extracting DNA, you require sodium acetate, SDS, proteinase K, phenyl to pH 8, and a mixture of phenyl chloroform amylic acid 25-24-1. But when you are extracting RNA, you need STS buffer, which contains SDS and tritone. You use trisol, chloroform, isopropanol, and the phenyl is adjusted to a pH of 5.
In the gel, the first lane is a 1000 base pairs DNA ladder, and it is shown in yellow that there are some peaks that could have been caused because the gel was pierced. The lane number 2 is the DNA sample, and lane 3 is the RNA sample. As shown in the red, there are proteins contamination in both lanes. In green, it is shown the peaks in the samples that could have been caused by salts contamination. Both samples were degraded, and it is visualized by the sweeping in the lane.
That our sample had an almost trustworthy pureness. It may be a little contaminated with proteins. Comparing the results of the electrophoresis and the ones obtained with the spectrophotometer, we can see that in both cases there is contamination of proteins. One of the samples is degraded and both of them could be a little contaminated with salts, as you can see at the top of the electrophoresis results. So in general, the spectrophotometer results and the electrophoresis results are equal. If the samples of DNA or RNA don't have a trustworthy pureness, then the results in the different reactions as PCR, enzyme restriction, and sequencing will be affected by all the contamination, avoiding a clear results for a lecture. If you have a low quantity of products because of a bad treatment of samples, you won't be able to do any reaction because there won't be enough DNA or If you have a low quantity of products because of a bad treatment of samples, you won't be able to do any reaction because there won't be enough DNA or RNA. As conclusion, when obtaining nucleic acid, it is important to look after the pellet forming the bottom of the microtube. It is better to leave supernatant in the microtube than lost sample. You have to be careful when mixing by inversion in order not to resuspend the pellet. It is also important that the chemical components used for purification have to be at the exact concentration and the exact pH, because the ion forces can affect the sample and degrade it.